Good morning, and thank you for joining us. I'm Emily Velasco, and I'm a science writer with Caltech's Office of Strategic Communications. It's my pleasure to welcome you to Conversations on COVID-19. This series is hosted by the Caltech Science Exchange and is designed to give us a chance to hear directly from the researchers in our community who are bringing insights and solutions to the COVID-19 pandemic. Today, I am joined by Emily Savala, Matt Cooper, and Alex Winnett. They are researchers from the lab of Rustam Ismagulov, Caltech's Ethel Wilson Bowles, and Robert Bowles, Professor of Chemistry and Chemical Engineering, and Director of the Jacobs Institute for Molecular Engineering for Medicine. Alex, Matt, and Emily, thanks for joining us. So I guess to get started today, for a little background, you all have been working on a COVID-19 study with the Pasadena Department of Public Health. So I was wondering if you could tell us a little bit about that study. Where did the idea come from and how does it work?
Sure, I can start us on that. So the big picture for the study is that we want to understand what's the natural history of SARS-CoV-2, which is the virus that causes COVID-19 infection, and how can we best develop interventions that can identify people early in the infection before they can transmit to others. And for me, the idea for the study sort of came from when I was working at the VA hospital. There was an outbreak in a nursing home, and the way that that outbreak was able to be quelled was by testing people at regular intervals to identify individuals who were asymptomatic, but infected and could be transmitting and perpetuating the outbreak. But the big question from that strategy, which was successful in that moment, was what is actually the right interval to test at? When will you catch people who are positive before they become symptomatic and transmit to others?
Okay, thank you.
So if I could add on to that, and how that works with the study in our laboratory, so we've enrolled these households where an individual in the household has already tested positive through a different testing site. And we look to also enroll their household members and household contacts before they have tested positive. So what we're doing is monitoring these households and observing any time transmission occurs to the household members. So that the way that works for us as researchers is they collect the samples every day in their homes, and they are couriered to Caltech with a medical courier. They're de-identified, so we don't know anything about where they came from or who they're identified with. We unpack the samples, inspect them, record all of the data, regularly screen all of those samples in our laboratory and quantify for positivity, and then also quantify the amount of virus in each of the samples. And then we report through study coordinators in our group to communicate with the study participants and the Pasadena Public Health Department in order to sort of communicate any positive transmissions.
Yeah.
Sure. I'm sorry, go ahead.
I was just going to say that one thing Emily touched on is the involvement of Pasadena Public Health. And while we're very focused on the lab side of this whole equation, leaders at the Pasadena Public Health, like Dr. Ying Go, who is the Pasadena Public Health Director, and Dr. Matt Feaster, who is a public health epidemiologist there, they were crucial in the early study design and getting this project off the ground. And they continue, and their entire team continues to help interface between the lab and the public to really keep this going and growing.
Great. So if I understand the study correctly, the idea is that if someone identifies as positive for having COVID, you look at the people that they live with, you study them, you study their samples over time, because the idea is that if you live with someone with COVID, you have a high chance of catching it. So say if you live with your parents and your brother comes down with COVID, you guys would be looking at the other members of the household to kind of watch their progress and see if they catch it. And if as they catch it, you can see in the results of their samples what's going on, right?
That's correct.
Exactly.
Okay. So what different kinds of outcomes can you imagine seeing? What, like from this study, depending on the results of what you come up with by the time it's done, like what are the consequences for how society responds to the pandemic?
Mhm. I can take this question. So originally the strategies for testing and diagnostics were kind of rolled out not based off of a lot of data, they're based off of a lot of predictions. So what our lab was able to do with this collaboration with the Pasadena Public Health Department is to gather that data that's missing, fill in the early part of the infection, and figure out what the best testing strategies actually are, whether that be a nasal swab, whether that be saliva, whether you need a high sensitivity test like PCR or a low sensitivity test like an antigen test in order to detect the amount of virus early on in an infection before somebody might even develop symptoms. So since we are actually capturing the samples before people become infected with SARS-CoV-2, we're able to start to answer those questions. So on more of like a population scale, we want to figure out the best testing strategy, what is most effective when people don't have symptoms, which will help people isolate earlier, inform when they have to isolate and reduce the spread to the communities, especially when they're thinking about traveling. You want to make sure that the test you have is the best one you can take to detect whether or not you're infected. And then on a more individual scale, the earlier detection provides more options for early treatment if you're developing a more serious illness, and can avoid possibly using later treatments that are more invasive. So better diagnostics helps in every aspect.
Right. So real quick, without going on too much of a tangent, you mentioned PCR and antigen testing. Just for folks at home who aren't familiar with those terms, can you like real briefly describe what they are and what the difference is?
So a lot of folks might be exposed to like a test that might take 10, 15 minutes, and that is an antigen test. So that detects sort of proteins on the surface of the virus, whereas PCR is a high sensitive test. It can detect very low concentrations of the RNA that's in a sample.
Thank you.
So those were ones that take maybe a day or two to get your results back.
Sure. So let's break it down a little bit. What role do each of you play in this research? I'm sure each of you has maybe a little bit of a different specialty and a different role to play here.
Yeah, I can lead us off here. I'm sure over the course of the hour, you'll get a pretty good idea of what we all do and where we fit into the bigger picture. But I also think it'd be really valuable to take a step back and highlight some of the unique skill sets and contributions from various members of the team. And I think it'll give everyone a better idea of what it takes to power a study like this and all the moving pieces that really have to come together to make it work. So Rustam Ismaghlov is the principal investigator and advisor of our lab in this project. And so really none of this happens without him. And working along him is Dr. Natasha Shelby, and she's the study administrator for the COVID-19 study, and she's also the research manager of our lab. And so she was a key leader in developing the design of the study and the logistics of making it happen. And once it got going, she really became the point person for overseeing the community interface. So Tasha sits very much in a unique position between the technical side of the lab and the Pasadena Public Health Department, our study coordinators, and even the participants in the study. And working with Tasha are Jessica Reyes and Noah Schlenker. And so Jess and Noah work closely with Pasadena Public Health to enroll new participants into the study. And they're the ones that are in daily contact with the participants in the study, answering all of their questions, motivating them and helping them get through the study. And then on the complete other end, on the lab side, we have people like Ana Romano, who is a research scientist in our lab. And she's been critical in validating and optimizing all of our quantitative detection methods, which you can imagine for a study like this is really important. And as the study has grown, we've had recent additions to the team, such as Alyssa Carter, who is a research technician in the lab, who already in a short period of time has gotten up to speed and is already handling a lot of the day-to-day operations in the lab. We also have really amazing undergraduate researchers in the lab, such as Jenny G, who was involved really early on in study design and has been taking the symptom data from the participants that they provide. And she's done some incredible data analysis and visualization that has been critical to link symptoms to viral load data. We also have other graduate students in the lab who are pushing new and exciting directions. As just one example, Michael Porter is one grad student in the lab expanding the study, and he's leading a project to quantify the amount of live virus throughout disease progression. And when we were trying to get this study off the ground, grad students like Jacob Barlow in the lab stepped up and created a database structure so that we could actually handle all these samples and keep track of them coming in each day. And I've also had the pleasure of working with him on some of the microbiome related aspects of this project. And finally, as the project grew and we needed to develop a higher throughput system to scale all of the lab operations, grad student in the lab, Reed Akana, built a new database and app for the project. And he's been doing some incredible computational modeling with data from this study and others. And so a long-winded way of that is we have a huge team and everyone is wearing many different hats. And where I fit into this picture, I was involved in the early study design and getting the project kind of off the ground. And since then, I've transitioned to focusing mainly on COVID-19 patients that have severe outcomes that are on ventilators in the ICU.
Okay, thank you. Alex, can you talk to us a little bit about your role?
Sure, yeah. Again, definitely want to emphasize that this is a massive team project. And we're all in such close communication, all virtually most of the time. And really, it's been the work of everybody to make this happen. But I've been involved in the early study design with the rest of the team, which includes supply acquisition, which during a pandemic can be pretty challenging, and developing the sample processing pipeline. And I'm really interested in the biology behind why people respond differently to SARS-CoV-2 infection, and also how we can decrease household transmission by tailoring public health interventions.
Great. And Emily?
Yes, thanks, Matt, for such a comprehensive view of the team. I really love the teamwork that we've been able to do. I love working with everybody. I have had the chance to start leading the swab and saliva comparison. So specifically, which sample type is better equipped to early detection. So I'm leading that analysis, and hopefully we'll have a publication soon to share with everybody. And I've also am a group biosafety coordinator in the lab alongside another graduate student, Mary Arastia, who helped with Lorianne Queenie at the AHS department, who's the Caltech biosafety coordinator or biosafety officer, to help develop all of our safety protocols and get everybody trained and making sure we all have safe SOPs to work with.
I'm sorry, what is an SOP?
Oh, a standard operating procedure. So make sure that when all of us are going individually, we do everything the same way. We have really strict entry and exit and decontamination and ways that keep us safe from the samples.
Okay, thank you. I'm going to have some questions about biosafety, but we'll come back to those. Mm-hmm. Okay, so you all have talked a little bit about developing this study and designing this study. I don't know that a lot of people know how a study gets developed. So can you talk a little bit about like, how do you go from like a nugget of an idea saying like, we want to study COVID patients to a full-blown study? Like, what's that process like?
I can jump in here first. So I think the first thing that we did was identify the clinical question or what needs to get answered. What's the gap in our knowledge? And then for us, that was understanding how does viral load change through the course of infection, and especially during that beginning part of the infection. So then the question becomes, how do you measure that? How do you capture that data? And this is, I think, where our study is sort of unique, and sort of the challenge behind this is that we want to catch people from before they're positive and they've been exposed and then become positive so that we can capture that data at the very beginning. And that's really challenging to do. We needed to think very carefully about what would be the right population from whom to enroll in this study to get that data. We thought about doing like just sort of a massive cohort of thousands of people and you just screen them all regularly, but that wouldn't be very efficient for finding people that go from negative to positive, which is how we eventually developed on a design that either follows an outbreak where after somebody in, for example, a nursing home becomes infected, everybody in the nursing home is at high risk and would be enrolled. The challenge with that is that you have to move really fast within the matter of hours to try and get everybody on board. And so that's maybe not as feasible. But eventually we landed on the case ascertained household transmission study design, which I think does that pretty effectively. And with that, we just want to we want to identify people who have very recently tested positive, because at that point, their household members are less likely to be infected yet when they would enroll in the study. And then we would be able to observe them becoming infected. And so this is where I think the benefit with Pasadena Public Health Department really, really comes into play, because we do still need to move fast and identify people early and be connected with them and let them know about the study and the opportunity to participate and answer these important questions about the pandemic. And they provide us with direct handoffs when they do their contact tracing, which is where they reach out to individuals who have recently tested positive to let them know about what they need to be doing, isolating, how they can protect their family, their household contacts, and who those contacts are that might have been exposed so they can follow up with them for infection control as well. They'll let them know about the study and pass them off to our study coordinators. So it's been extremely valuable to be connected with the public health department in that way.
All right, thank you.
Yeah, just to add on real quick to what Alex was saying, I don't think we can emphasize enough just how hard it is to catch that early part of infection in individuals. And we knew this was a problem from the start. And so once we locked in on this idea of how to do it, that partnership with Pasadena Public Health was really critical. And so without not just the partnership, but without them really working to get those quick handoffs from clinicians that immediately identify someone as positive and then transferring them over to our study coordinators, we would not be able to capture the data that we do.
Yeah, that makes sense.
And then I guess on a more specific side, and from the laboratory, there's a lot we had to do to develop our workspace. We needed to validate that we could use the CDC assay on the samples. So we want to make sure that we can quantify everything properly, had a lot of control experiments to run. And although our lab has had, the three of us have had a lot of experience in the Izmailov lab handling participant samples, we haven't handled anything on this scale. So we had to do a lot of work in order to handle the number of samples we do every day and make sure that we have, as Alex said, even just getting supplies to do RNA extractions at this time is non-trivial. It's a lot of extra work just making sure we can get the equipment, get the supplies that we need to run this number of experiments. So that really informed some of those early questions about, it'd be great if we could monitor thousands of people every day, but we really don't have the throughput and the capabilities to do that. So we had to think very carefully about how we select the households to make sure that we're selecting ones that fit a lot of our study criteria to get us the best chance at the data we need to answer those questions as early as we can.
Yeah, thank you. So to kind of drill down a little bit here, let's talk about the virus itself, or rather the viral transmission. We've heard the term today, viral load. And I think for people who aren't familiar with this research, that might be a little bit of a mystery what viral load is. So maybe could you guys talk about what viral load is and biologically speaking, what happens when someone contracts the COVID virus? What does that look to you guys as you monitor that person?
Yeah, that's a great question. There are tons of terms sort of being thrown around related to the pandemic, and we definitely want to make sure that we're all on the same page with what things mean. So in terms of viral load, the background here is that we know SARS-CoV-2 is a virus, and it's specifically an RNA virus, meaning that it uses RNA as opposed to DNA to encode the genes that it needs to replicate itself. So when a SARS-CoV-2 virus meets and enters a host cell that it's compatible with, it will use that RNA to make proteins and sort of reprogram that host cell to produce more of itself. And the cell does interesting defenses to try and prevent that. But if the virus is able to continue to replicate, the cell will then pop, and those viruses will then go on to infect other cells. So over time, what you'll see is that you start potentially with one virus, and then it replicates very quickly in the host. And so when we say viral load, what we mean, that's a measure specifically of the amount of RNA from the virus that we're seeing in these samples, which early in the infection will be very low, but it will increase at some rate.
Okay, so it's a measure of how many viruses a person is carrying at a given time, right?
Exactly, yeah.
Okay, thank you.
And then if I could add a little bit on that from the lab side. So we collect participant samples and we measure a couple different things from each sample. One of them is just a marker that the sample is in fact like a human sample. It's more of like a quality control thing as well. So we want to make sure that there is human material captured from the human sample. And then we check that first and then measure the amount of SARS-CoV-2 RNA. And then when it comes to looking at whether it comes from a nasal swab or a saliva sample, comparing the amount of virus detected from the same person collected at the same time, and how much virus is in each part of their sort of respiratory system. And then I guess, so when we talk about viral load and how that can actually help us answer some of these questions, like what test do you use? So each types of test have a different limit of detection, and they'll be able to say, a really highly sensitive test, you can detect very few copies of the virus or low viral loads. And a lower sensitivity test requires a much higher concentration, like orders of magnitude more virus or viral load than those samples. So as we are building these curves and measuring how much of the virus you have in each of your nose or your mouth, we're able to infer which diagnostic test would have picked you up at how many days you are along the infection. And that's something we're really interested in, before you have symptoms, sort of what tests are detectable, because a lot of people don't get tested until they're symptomatic.
Right, and it's important to know what kind of test to use, because if you use the wrong test, it might not even show that someone's carrying the virus, right?
Mm-hmm. Absolutely, yeah.
Okay. So this question is for you, Emily, because you talked a little bit about biosafety earlier. What is it like to work in the lab and handle samples carrying COVID? I mean, COVID, it can be deadly for people. So what is it like to handle a virus like that? And talk to us a little bit about what kind of safety measures you take when you're working in the lab.
So I should say, firstly, almost all of the samples that come into our lab are inactivated already. So they're in a preservation solution. So that already makes it a lot safer for us to handle, because the chances that the virus is able to then infect us is incredibly low. So already we have that sort of safeguard in place. But we really worked closely before, in the early stages of the planning, to work with Lorianne, the biosafety officer at Caltech, to develop these protocols and figure out what sort of PPE we need, our personal protective equipment, which is probably a term most people are familiar with nowadays, as you wear masks every day. So her help has really been invaluable, making sure that we're working safely. So making sure we have a buffer to inactivate the samples with before they get to our lab. And once they get to our lab, we have protocols on how we open the packages. But before that, I should say, we have limited capacity. So we only have usually one person working in each part of the lab at a given time, which requires a lot of coordination and communication among people. We have a lot of training, a lot of internal protocols and communication between not only those of us on the project, but there's many people in the lab who don't work with COVID samples at all. And we want to make sure they know what's going on and that they feel safe working in a room next to us. And when we actually go in to handle the samples, we wear 2 lab coats, 2 pairs of gloves. We have sleeve covers that come up to here over our lab coats. And we wear respirators, which are like a big helmet with our own HEPA filter. And it looks kind of like a spacesuit. So we are very aware as we're going in that what we're about to open up is much more hazardous than things that we're used to. So I think all of us feel very safe working with those samples because of how routine everything is now. We had a very strict bar at the very beginning for how we were going to approach this. And now we've done it so many times, and like I trust that the person in front of me or person behind me has followed all those procedures. I should say we also have like a good buddy system, because like our samples come to our lab 7 days a week, so we kind of have staggered sort of schedule for who goes in on which day. And sometimes you're the only one in the lab, so we want to make sure that somebody knows that you're about to go in there. So we have like a pretty strict system with that as well. So.
Thank you. So I'm thinking about monitoring people as they go through the study, and thinking about getting samples from someone who's living with someone who has COVID, and the person you're getting samples from doesn't have it yet. I feel like I would be a little nervous for them, watching them go day by day living with someone who has COVID. Like, they might get infected, and you don't know what the outcome is going to be. Do you guys get nervous for the participants as you monitor them?
Yeah, definitely. And I feel like, especially lately, I get a little extra nervous since I'm also working with samples from ICU patients with COVID. And so to see the samples that we receive from people that don't report any symptoms, to some that report basically a mild cold, to others that don't recover from this, it lets you see the spectrum of what can happen. But what I should say is, on the lab side, we're very removed from this all. We never know these people's names. We don't really know much about them. We just get tubes with IDs on them, and we go about our lab work. While people like Tasha, Jess, and Noah on the study coordinator side are interacting with these people every single day. And so I can't imagine what it's like for them to see the science and to see the viral loads going up and the transmissions occurring, and also speaking to these people that are often very frightened.
Sure. This question is for all of you. Has working on this study changed the way you view the world around you during a pandemic? I think a lot of people, the pandemic for a lot of people is sort of, a conceptual thing, and it just means, stay at home. But you guys are seeing some of the real effects. So has it changed the way you view the world, the outside world today?
I think we're all pretty cognizant of what diagnostic tests can and can't do, and what they do and don't mean. And so when I have friends and family that go, oh, I just got a test, it's negative, I'm fine, I think I'm probably a lot more weary than I would have been in the past, because I'm thinking about, well, what day post-exposure and what type of test did you do and how did they handle it? Because that will all influence it. So it's definitely maybe made me a little more paranoid.
Okay. Alex or Emily, did you have anything you wanted to add?
You can go, Emily.
Oh, sure. Yeah. I mean, I exactly what Matt said. I have a lot of follow-up questions anytime somebody says I tested negative. I'd also say I'm a little bit more aware every time I see somebody wearing a mask that doesn't cover their nose, because all I see immediately is just these viral load plots that are just like seared into my brain of like, I know how much virus you could have in your nose even without any symptoms. And that's something that like I cross the street if I'm walking near somebody who has their mask on improperly. And I should also say that's something that's very important about biosafety is PPE is excellent, but only if you know how to wear it properly and disinfect things afterwards. So if you can take it off properly and wear it properly, that's what protects you. So even people can wear masks, but if they're not wearing it properly, I get a little anxious.
Right. Alex?
Yeah, I would add, I mean, the pandemic has been so incredibly hard for, I think, everybody. And so for, I think, some of us on the lab team, that means potentially being in a small room, processing samples for a long time, wearing all of this PPE, thinking about the participants that we have in the study, and really confronting what's happening in the world, even inside this little room by yourself. But I think that working on this study, what that's really meant for me it's really underscored that we are all in this together, that even if we're remote or we never know these participants, this pandemic has affected every single person. And I don't know if there's many things in life that have that type of effect so acutely as well. And with that, I mean, we all want this pandemic to end. And so I've also really appreciated the opportunity to be able to work on something that might contribute data to make this a little bit less tragic for everybody.
Yeah, definitely. So last question, I think, is where does this research go from here? You all have collected a lot of samples now, and I understand that you're working on a couple of papers. So what's next? Where do you go from this point forward?
Yeah, I can start here too. So people have very different responses to SARS-CoV-2 infection, and understanding why somebody will become really sick versus just have a cold versus not display any symptoms at all is something that's really important, so that we know how to best help different people. And I think that that's often related to the host response. We know that there are certain associated conditions that put you at higher risk for severe disease, and we're starting to create those links as to why that's the case. So I'm really interested in looking at our samples, not just the virus, but also what we can glean from that about how the host is responding and how the infection might look different in people that respond differently to the virus. Matt mentioned earlier that Michael Porter in our lab is interested in looking at the amount of infectious virus in the samples, because even if the virus is replicating, even if we detect, RNA or something like that, that doesn't necessarily mean that somebody could transmit the virus. You might have seen reports that people can remain positive by tests for weeks after they were initially infected. But previous studies that have been done have shown that it's very unlikely that they would have actual infectious virus at that point. But no study has been able to show it before symptom onset and whether somebody's infected then. And so the argument has been made that, oh, when viral loads are so low, there's no infectious virus. But we don't know that that's the case early in the infection. Also, with the emergence of variants, Ana Romano in the lab has really taken the lead on, we're sequencing the virus in these samples to understand what variants people have and how that might influence the disease progression and the viral loads that we're seeing. And then how does the data that we have potentially affect population level health? And so Rita Khanna, one of the other graduate students in the lab, is using some really fantastic modeling skills to apply what we're seeing in our samples to policy implications and how the pandemic would progress if we attempt different sorts of strategies. Mm-hmm. And then we also have Jenny G that Matt mentioned earlier, one of the undergraduates that's looking at how symptoms are related to all of this and also pushing an analysis of what are the factors that modulate household transmission and how can we sort of tweak different things through public health intervention to reduce the risk that we'll see people become infected in the same household.
Right. All right. Thank you. I think in the interest of time, we can move on to some Q&A questions now. I think we've gotten quite a few from audience members. So let me take a look and see what we've got here. And you all can just answer these as whatever seems appropriate for each of you. The first question we have is from Robert Whiting. I hope I'm pronouncing his last name right. And
he asked, before we jump into the questions, I think Matt has a really interesting analysis that we should mention too.
I'm sorry, Matt.
Oh, no, just I want to mention that, everyone's starting to get vaccinated and pretty soon a large chunk of the population will be vaccinated. And one collective effort by the lab is to understand if people can still transmit if they have the vaccine and what the transmission looks like and viral load looks like in those individuals. And then even beyond COVID, our lab is really trying to utilize all of these samples we've collected. We have thousands of them now. And so we think these will help us get a much better understanding of people's microbiomes in both healthy and disease states. And so people like Jacob Barlow and myself are really interested in pushing that direction further.
Great, thank you, Matt. So Robert Whiting asks, is there a good book that any of you could recommend for someone who's not a virologist? Is there anything out there that gives a good overview of this topic in general?
I mean, about specific to COVID, no, but I know there's some really interesting books about the sort of 1918 influenza pandemic, which are really interesting.
Mhm.
And other, I would just say, thinking about any books that look towards, the SARS pandemic or other things like that. So I think historically, those would be the places I would start in terms of trying to get an understanding of respiratory pandemics. But I'm sure there's going to be some really great books that are coming out here soon.
Sure. We have a question from, it looks like Renee Katzenstein, who asks, how large is the study? How many cases are in your sample size at this point? And what's the geographic area for your study?
Yeah, I can take this one. So I think to date, we have had over 160 participants in the study from over 50 households. And like Emily was saying about the high volume of processing samples, people are providing both nasal swabs and saliva twice per day. So each person provides 4 samples per day. And so we've received over 5,000 respiratory samples. And these participants hail from Los Angeles County, Pasadena, and Long Beach.
Okay. So I've got a question here. Looks like from Kathy, who says, are you all vaccinated? You don't have to answer that if you don't want to, but are you all vaccinated? And what are your thoughts on people who think it's more dangerous to get vaccinated than to catch COVID?
I'll say that I've received my first dose and I'm very excited to get my second dose. And to me, I would say, of course, talk to your doctor. And if you have some condition that precludes you from getting the vaccine, definitely listen to them. But if you're otherwise healthy and just on the fence, I would take maybe feeling a little run down for a day or two and know that I'm protecting myself and, more importantly, most likely helping those around me. And so I would definitely, if you're on the fence, take it.
Okay, thanks.
I'd also,
go
to
ahead.
that point, mention, I think that it's fair and it's fine and it's not a bad thing to have healthy skepticism about anything. And so I think that it's good to be asking these questions and doing the homework and seeing what's there. I do think that there's a lot of data about vaccines because I think everybody's really excited about them and wants to know, is this going to be our ticket out of the pandemic? So I appreciate this question a lot because I feel like sometimes we're so excited about vaccines that we maybe don't take a moment to recognize that people are having these questions, and they are really important questions. I've worked at vaccine distribution sites, the megapods, and specifically the one in Northridge. And if it's any consolation, we monitor people for the time after they've gotten the vaccine. And at my site, we haven't seen anybody with severe reaction. And actually what I hear most of the time from people that are coming back for their second dose is, I had a sore arm, or I maybe felt a little bit like a cold. But generally, I think people are really ecstatic to be getting the vaccine because that means protection from something that is probably a lot worse.
Sure. So we have a question here, and it asks about children. They want to know if there are any children that have been participants in your study, and if one of you can talk a little bit about infections in children and whether it makes you wonder if reopening elementary schools could further spread the virus.
I mean, that's a great question. Yes, we do enroll children in our study. I think, Alex, correct me if I'm wrong, I think as long as 6 years old?
Yep, 6 and up.
Yep, 6 years old and older. So we've had definitely individuals in the elementary school age and high school age in our study. And I think this is a really prudent question. And I'd say I'm very excited to hear that so many educators have been prioritized to get the vaccines. I think that's absolutely critical. But we also need to be very aware of the other practices, such as mask wearing and hand washing and distancing, especially when people are eating. So I'd say that just because schools are opening up, we have to be, I'd say, extra careful. And I think that, I mean, none of us are policymakers, but I would just say, if you're in that situation or if you know people in that situation, definitely emphasize how much we know about hand washing and mask wearing and distancing and how that can help reduce your chances of getting transmission.
Yep. Okay, we have a question from Mike Harris, who asks, do you think that there's a gap between the understanding that you've gained from your research and what is typically reported in the media?
This is a very interesting question. I think everything's a balance, right, between simplicity and nuance. This webinar is 45 minutes, but this is a topic that could go on for days probably. And so I think that those trade-offs get made in the media, and we're trying to figure out what are the important nuances that really do need to be captured succinctly to make sure that we're making the right decisions in terms of policies and practices to decrease transmission.
Yep. Yeah, communication is vital. And that's why we're doing this today, right? Okay, we have another question from Stephen Johnson, who wants to know, once he's been vaccinated, what he could expect from a COVID antibody test, which I assume he's asking, like, if he's been vaccinated and he gets a typical COVID test, is he going to show that he has antibodies for COVID?
That's a great question. I don't know if I know the answer to that one.
I think
Yeah, that's fair.
Yes, I think typically you will develop an antibody response. And the hope is that that antibody would be protective against the next time you're exposed to the virus.
Yeah.
There were some studies that showed potentially after natural infection that antibody levels wane over time. And that's been shown with other respiratory viruses as well. And I'm not super familiar with whether that's the case with vaccine, but there have been discussions of booster doses either to bump that up or to capture other variants that might not be as well protected by antibodies that are generated from the first few doses of the vaccine.
Okay. And on that note, we have a question from Jessica Crick, who wants to know if your study continues to monitor participants even after they've gotten vaccinated. Is that data that you guys collect? If you find out a participant has been vaccinated, do you continue to collect data on them? And if so, how does that affect the data that you're seeing?
I think it's more of an enrollment question. So we know before we start collecting any samples if they've been vaccinated or not. And I don't think we've encountered very many people who have been vaccinated yet, but this is something that we are asking people now and sort of including in our data analysis later on. So yes, we're going to still enroll people if they've been vaccinated, if they meet all of our other study criteria. We have a lot of it, in order to keep our sample numbers reasonable in the laboratory. But short answer is yes, we will be.
Okay, thank you.
We do ask questions after people have participated in the study. We do a post-enrollment questionnaire about what happened in the last 2 weeks. And as Matt was saying, it's a stressful time for the participants who are in a house where they might be at risk or other people that they live with might be at risk of COVID. And so I feel like they've formed pretty strong connections with our study coordinators. And so I think there is some contact continuing with the study coordinators and participants. And I think most people say that they would be interested in follow-up studies after participating in this one.
Okay. And I think we have time for one more question from the audience. So let me see here what we've got. All right, I guess so. Philip Appleton asks, at what level of viral load would you consider someone to have a full-blown infection? I don't know what full-blown means, but I guess he's maybe asking, at what point would you say someone is sick with COVID based on their viral load?
I feel like that's almost a philosophical question. At least I don't know if I could pin a point, because there are some people we catch very early, and I would say, well, they were probably sick with the SARS-CoV-2 infection before we caught them. But they're definitely a few days later still very much sick. So at least from my perspective, I don't think I could at least define it.
Right. I mean, I guess it really just takes one virus to be infected, right?
Yeah, and I mean, for other viruses and bacterial pathogens, we do try to characterize how much it does take to get into your body to typically cause an infection. But to my knowledge, I don't think we have that number for SARS-CoV-2.
Okay. Thank you, Matt. I think we're out of time on questions. Well, I want to thank you all for participating today. This has been great. And for, thanks for joining us. And for everyone at home, thank you for joining us as well. For folks who are interested in maybe being participants in the study or even volunteering to assist with the study, if you'd like more information on the study being conducted by the Ismagulov Lab, you can visit covid-study.caltech.edu. That's covid-study.caltech.edu. In the meantime, if you're interested in exploring more of the science behind viruses and COVID-19, please check out the Caltech Science Exchange. The site launched last spring to provide trustworthy answers and clear explanations on topics in science and technology. You can find it at scienceexchange.caltech.edu. And thank you again, everyone, for participating today.